Okay, so let's look here. Okay, what do you think that is? That's microvilli. To me, microvilli looks like somebody took a highlighter and outlined the apical surface of the cell. It's really, really neat. Somebody neurotic took a highlighter and outlined the apical surface of the cell. But cilia are really, really messy. So let me show you cilia in a slide that you probably haven't seen before. Can you see? Cilia look kind of like bedhead on the surface of the cell. They're not nice and tidy, okay? So you can tell the difference between cilia and microvilli. Now, you do not want to be the person who only looks in one area, right? You want to like get sort of a more global look. So let's look at a couple of different tissues and see if you guys can tell me whether you think you're seeing cilia or microvilli. What are you seeing here? Cilia. Yeah, you're seeing cilia right there. Now this one, look multiple places. Cilia. That is microvilli. Okay, this is just mucus that you're seeing on the surface. This is not cilia that you're seeing on the surface. So you got this sort of highlighter going along there. How about this one? That one's a hard one. That one's actually microvilli, but you wouldn't really know it. Okay, so let's look at a couple.
First one where we're looking for that.
Here? Okay, but you also just have to get used to looking at it because, guys, you can actually see individual hairs when you are looking at cilia. You do not just see the suggestion of something messy on the surface. Go ahead.
Will the microvilli always be a different and or darker color?
It's usually either darker or lighter depending on, it's usually distinct from the cell itself is what it is. That one is microvilli, right? Okay, so how about this one?
Cilia.
That one's cilia. Okay, so can you see that if you practice enough, you start getting confident at it? Initially, it's like, I'm not quite sure, especially if there's mucus on the surface. But after you get used to looking at several of them, you can see that this actually looks like bedhead, right? It looks really messy. And you can see individual little hair-like structures.
Does all tissue have either one or the other? Can the tissues have both? 2 questions.
Okay, there is no tissue. I'm averse to saying never because, you know, I've been a biologist for a while. I've never run into a situation in which a tissue has both. Okay, so it will be one or the other, especially with the tissues that you're looking at. I'm not going to use a tissue that has both because I've never seen one, couldn't find one. But even if there was, I wouldn't choose that for an image for you. Does that make sense?
Like mucosal linings?
What?
Cilia, isn't it more along like mucosal linings?
It's any. No, they're both in mucosal linings. Okay. Yeah. You guys okay? So we're going to practice it a little bit more as we go through this other ciliated tissues so that you'll sort of get used to it. Let me show you a resource as well. This site right here, which is, you can get to it from, let me show you where you can get to it. So this is melanieb.com, and this is the links page of melanieb.com. Okay. Look right here under histology. There's several links to different places to practice histology. This first one is the college that I used to teach at before I started teaching here. And it has printable flashcards that are labeled on one side and not labeled on the other. So you can use these to sort of get used to it. So do you see this one? Even though they're nice and neat, can you see that they're not just a highlighter, right? Right, that's cilia. So these are printable and you can fold them over and they're labeled on one side and not labeled on the other. That makes sense? Okay, so that's cilia versus microvilli. Oh, let me show you cilia in action too because it's kind of cool. So here is what, we're going to look at cilia in a little bit more detail in just a second. But when you look at cilia from a surface view, okay, let me show you what it does.
One defense mechanism
We don't want to
mammals
listen
use
to
to
it.
prevent invasion by pathogens is the ciliated epithelium that lines the respiratory tract.
Yeah, it's kind of cool, right?
The cilia sweep mucus and particles into the throat, where they are swallowed or coughed out.
They call that the mucus elevator in your throat. That's sexy, isn't it? You go use that at home, you're definitely getting lucky. Okay, so see, where were we? Okay, so that's all of the simple tissues, and then we'll come back. Oh, let me tell you something about the simple tissues as well, or all of the tissues. One of the things that sometimes your lab instructors don't really focus on, but I do, is that you need to know where a tissue occurs. You need to know body locations for tissues. So with body locations, what you need to do is play Magic School Bus verbally with me to get me to the right place. So let's talk about where you would find a couple of different tissues. So let's talk about this one right here. So this tissue, do you know what it is? It's simple columnar. Simple columnar. And by the way, guys, here's something that I will do for you whenever you guys are taking tests or quizzes. What I will try to do is point you to the area that's the best indication of what tissue it is, because he just said pseudostratified. Because if you start looking here, because it's under cell division, it looks like it might be pseudostratified, but there's a couple of ways to tell that it isn't. So I will point you to the area that I think is safest to look at. Never think I am trying to tank you. I'm not. I'm trying to help you because anatomy is hard enough. So if we look over here, can everybody agree that this is simple columnar epithelium, right? So then I would ask you a body location for this tissue, and you would say what? You would say what? You have to be far more specific, because my Magic School Bus does not know what digestive tract means. Nope, not stomach. The lining of the small intestine. Yes, you can't even just say small intestine, because we know that organs are composed of multiple different tissues, right? And if I went into the small wall of the small intestine, I would run into both connective tissue and smooth muscle. So what you are going to do is to memorize a really safe body location for each of these tissues, instead of all of them, which means that it's going to be a fill-in-the-blank question, right? Does that make sense? Because if it was a multiple choice question, I would have to have you memorize every body location for the tissue. But if it's fill-in-the-blank, I can say, name a body location for that tissue, okay? So now let's do the other one. What about this tissue?
Lining of the small intestine.
Yes, lining of the small intestine would be great.
Okay.
How about this one? What is the tissue? And then give me a body location. Or this is the same tissue. Ciliated simple columnar epithelium, a body location. Yeah, lining of the uterine tubes or fallopian tubes. And notice that she said lining, right? Because obviously they're going to be made of multiple different kinds of tissues, so you have to get them to the right place. So usually with epithelial tissues, you're either going to say lining or superficial surface or something like that so that you get to the right place. Does that make sense? Okay. Now, in lab, they don't concentrate on that, but the reason I do is because I'm going to be referring back to that all semester long. So instead of you just looking at the fallopian tubes or uterine tubes, when we get there, I will say, do you remember what tissue is lining the uterine tubes and what does that structure have to do with the function, right? Does that make sense? Okay, so we're going to sort of reinforce that all along. And this is what tissue? Here? Simple cuboidal epithelium. Anybody know a body location? Nope. Nope. Lining kidney tubules. Okay, you might not know what kidney tubules are. You will this semester, and you'll continue to talk about it in physiology. It's these really, really curved tubes within the kidney that allow for direct exchange between blood capillaries and urine, right? This is actually, these tubes are really involved in cleansing your blood and getting stuff out. So this is kidney tubules. And then the last one, this tissue right here, give me a body location for it. There's several. What? Alveoli of the lungs. What else? Yeah, capillaries, lining capillaries. Yeah, lining your capillaries or all of your blood vessels, really. Is that it? Okay? So that might take a little practice. What? Simple squamous epithelium. Yeah, exactly. You can say squamous. I just say squamous. I don't even know what's right. Okay, so those are your simple tissues. Now let's move to the stratified tissues. Ready? Okay, so what makes something stratified? You know, just verbalize it for me. More than one layer of cells from the apical surface to the basement membrane, right? Okay, so let's look at a couple of different tissues that we've got here. So this is, of course, a stratified tissue because you, for me, what I use to indicate that there's more than one layer is nuclei, yeah? Can't see nuclei in every cell, but you can see them in most cells, right? And so I've got several different layers of nuclei as I move from the apical surface to the basal surface. Now, this is, of course, stratified squamous epithelium, but not because all of the cells look squamous, because at the bottom they don't, right? but this one is labeled or named for the shape of the cells at the apical surface, and this is stratified squamous epithelium. Now, there are two categories of stratified squamous epithelium. They're really similar to one another. This is moist or non-keratinized stratified squamous epithelium. Can you guys tell me which category of epithelial tissue is going to be more protective? Simple or stratified? Yeah. So you're going to find this at places that have abrasion, yeah, but don't have to worry about water loss because they have a source of moisture. Okay. So can anybody tell me a couple of locations for this tissue? Lining the mouth, yeah. Lining the distal end of the digestive tract, the rectum and the anus, right? Lining the vagina, right? That's where you find this tissue. Is that good? Okay. So this is moist or non-keratinized stratified squamous epithelium. Now I want you to pay attention to distinguishing that one from keratinized stratified squamous epithelium, because what keratinized is this plus a big layer of dead cells on top. Because the cells on top are dead, you won't see nuclei, okay? Dead cells filled with a waterproofing protein called keratin. Okay, so although it doesn't look like it in this image, what I want to tell you is this picture that you're looking at right here is really just what looks like moist, it's not called moist, but a keratinized stratified squamous epithelium with nuclei up to here, and then what do you see on the top? A big old pile of trash is what you see on the top, right? Waterproofing trash. So where do you find this tissue? You find this tissue in locations that are subjected to abrasion and have to be concerned about water loss. Yeah? So be more specific. Epidermis, if you know that word, or if you didn't know epidermis, you could use something directional to get me to the right portion of the skin. Superficial surface of the skin, right? Does that make sense? Okay, so now let's look at a couple and see if you guys are comfortable with knowing which one this is. What is, no, let's not do that one. That one's a little confusing. How about this one? What is this? Nope. This is moist. And how can you tell? So let's get used to like the discomfort. How can you tell this is moist? Nuclei all the way to the top. Does that make sense? Nuclei all the way to the top. So let's look at one that is keratinized. You ready? Okay.
So do you say moist or can you say non-keratinized?
Either one is correct. Moist or non-keratinized. So let's look at this one. Do you see? Nuclei all the way up here. Yeah? Plus a bunch of trash on top. Yeah, this would be like the palms of your hands or the soles of your feet where it's really, really thick on the surface. Does that make sense? So moist and keratinized stratified squamous epithelium. Good? Okay, they're both protective. One of them doesn't have to worry about water loss. Everybody good? Okay, so you can practice that. You need to look for the nuclei is what you need to look for. It's not so much the thickness, it's like whether there is the presence of nuclei on the surface. Okay, so let's see. Do one more. Do I have another one on there? No, I have another one. No, that one confuses people. This one's hard. Look at it carefully before you identify it. What do you think? It's moist. Yeah? Initially, because the texture changes a little bit, you would think keratinized, but then what made you revise your decision? Nuclei all the way to the surface, right? So again, like anything else, histology was harder last week than it is this week, right? You got a little bit more exposure and a little bit of the struggle of getting through it, and you've seen more slides, so you get better with it. I'm going to go ahead and tell you that happens all throughout anatomy. Please expect to be overwhelmed, and then things clarify a little bit, and then you feel like you can do it. And it's going to happen every damn time, and sometimes it's going to be really, really uncomfortable. Like next week when you guys start doing the skull, you might cry. Somebody will probably cry next week. But it's okay, right? Because what is the first, this is what I tell my 12-year-old all the time, and I realize I'm not your mom. But I do all the time. I'm like, what's the first step to being really good at something? And he's like, sucking at it. And I'm like, yep, got to be prepared to suck before you rock. So get prepared to suck and then rock. Okay.
Wait, question. What's this stuff on the far right then, where it's like the hollow circles? They don't look like nuclei because they're not darker?
They are nuclei.
Oh,
It's a different staining technique.
Oh, okay.
Yeah, just different staining technique. Okay, so that is stratified squamous epithelium, moist and keratinized, or non-keratinized and keratinized. And then let's look at stratified cuboidal epithelium. Stratified cuboidal epithelium is not very hard, I don't think. Okay. Now, this one you can't really see the boundaries of the cells very well, but you see the suggestion that they're about as wide as they are tall, right? Okay. And you see the nice round nuclei that can kind of indicate to you that that's what you're looking at. Can you see the suggestion of stratification? Yeah, right? They look like stacked on top of one another. This is stratified cuboidal epithelium, but does it surprise you that so is that? Right? Or this one? Right? Stratified cuboidal epithelium. And that's not bad, right? Okay. Where do you find this one? Anybody know? What? In the body, for sure. Yes, he is. Right. No, it's actually found lining the ducts, not quack quack, but the other kind, of lots and lots of glands. So it's found like forming that. Okay, so you're gonna have to practice that a little bit. Go ahead. Well, considering that you haven't learned the glands yet, I'm not going to make you use words that you don't know the meanings of yet, right? Because that's just BS, right? Later on, when you know the meanings of things, then you're responsible for those words. Okay, good? Okay, lining the ducts of some glands. So that's stratified cuboidal epithelium. And then this relatively rare tissue right here is stratified columnar epithelium. It's easy to confuse with pseudostratified, but pseudostratified always has what on the apical surface? Always has cilia, right? So you use these sort of clarifying things. If it was pseudostratified, it would be ciliated, and this one is very obviously not ciliated, right? Okay, so this is stratified columnar epithelium. It has the suggestion of 2 layers of oval nuclei. The cells look taller than they are wide, right? So stratified columnar epithelium. And you only find this one place in the body that you will care about or know about, and that's where? Yeah, it's really only the distal end of the male urethra. Males and females, of course, both have urethras, right? But females, the urinary bladder leads to the external urethral orifice, and it's only like an 1.5 inches long in women. Okay, short urethra. We'll talk about its tissue type in just a second. In males, the urethra has to go from the under the urinary bladder all the way through the penis through to the external urethral orifice. And so on average in males, the urethra is around 8 inches long. I am not telling you that a penis is on average 8 inches long because I was literally not born yesterday. But if you go all the way up to the urinary bladder, right? And of course, the male urethra has double functionality compared to the female urethra. The female urethra, on a good day, only ever has urinary functionality, right? The male urethra, on a good day, would have double functionality, right? It would have urinary functionality, right? And then it would also have ejaculatory functionality for the reproductive system. And that's the whole idea of the change of tissue type at the distal 1/3. And we'll see its functionality when we get to the reproductive system. So it changes tissue type at the distal 1/3. And I don't know of another place in which that tissue occurs. Go ahead.
Are there multiple types of tissue in the male reproductive system or urethra?
Yes. So it's lining.
Oh, the lining.
It's going to be lining the male urethra. Yeah, lining the male urethra. And specifically, although I won't ding you on it, it's lining the distal portion of the male urethra. I won't ask you to say distal 1/3 yet. When we get to the reproductive system, you will say, okay, I know exactly where it is, right? I know. All right, good? Okay. All right, so that's stratified columnar epithelium. And then we get to transitional epithelium, which somebody didn't get the memo for how to name things, right? Because it breaks the 3-word naming things. I don't know. Okay, and this is transitional epithelium. Transitional epithelium is found lining almost the entire urinary tract. The ureters, yeah, going from the kidneys down, the urinary bladder, and the entire female urethra, right? And the proximal portion of the male urethra. And the primary characteristic of this tissue is that it's stratified, even though it's not in the name, it is stratified, okay? And that it looks really different when it's empty versus stretched. Like imagine this is in the urinary bladder for me, okay? And this is in an empty urinary bladder. When it's empty, the cells at the apical surface are domed, right? And when it's stretched, when the urinary bladder is full, can anybody sort of imagine what this would look like if it was stretched?
Flatly, more like
Yeah, yeah. What it might look like a what? like a moist stratified squamous epithelium, which is why I never ask you it stretched, because that's mean. Okay, but it does look like moist stratified squamous epithelium when it's stretched. So this is transitional epithelium, and it's found lining almost the entire urinary tract. Okay, after the kidneys. It's not in the kidneys, but after the kidneys. Good? Okay, so that's transitional. And then our last tissue is, our last epithelial tissue is pseudostratified. And what does pseudo imply to you? Yeah, false. So let's look at this tissue and make sure that you can tell first why it's called pseudostratified. The reason it's called pseudostratified is that, of course, it looks stratified, right? It does look like it's layered, but it's actually not, because what you're actually seeing is that all of the cells are stuck to the basement membrane, but some of them are taller than the others. So we end up with a tissue that looks like this, right? But it's false stratification, okay? Pseudostratified. Now, the cells that reach the apical surface, they will be ciliated. These cells are not ciliated because what the hell would the point of that be, right? That's a waste of protein synthesis to ciliate anything down there, right? But the cells that reach the apical surface will be ciliated. In addition, this tissue often has visible goblet cells. We'll talk about goblet cells more in just a second. So how do you name this tissue? Well, nobody can agree on what order the words go in, so I don't really care. I usually like for you to use all four because it tells you everything you need to know. Ciliated, yeah, pseudostratified, columnar, epithelium, yeah. I don't care what order those go in because nobody is particularly consistent about the order, right? But if you want to be really safe, that's probably what you would put, is all four things: ciliated, pseudostratified, columnar, epithelium. Sometimes people say pseudostratified, ciliated, meh. Potato, potato. Huh? What?
Always columnar, right?
Yeah, they're always columnar. Yeah, the cells are relatively tall and skinny. Although not all of them look like it, but it does look more columnar than anything else, right? So this is ciliated, pseudostratified, columnar epithelium. Where do you find this tissue? Well, it's got two things that kind of tell you structure-function relationship. The first one is the cilia, of course, and the cilia here are going to be waving this coordinated mechanism. And the second one is the goblet cells. You find this lining the upper portions of the respiratory system, right? And so what are the cilia for there?
Catching dirt.
Yeah, so you're constantly breathing in particulates, right? Especially living here, right? And what the cilia do is they wave up to keep it from going down to the respiratory surfaces of your lungs. And what do the goblet cells do?
Provide music.
Yes, the goblet cells secrete mucus. So when they wave up and then you, nobody wants to know what you do with it. I don't care if you swallow it, but let's not talk about it, okay? Swallow it, spit it, whatever the hell. Don't do it in front of your spouse. That's all I'm saying. Right? Just do not. Nobody likes it. Grounds for divorce. That and thermostats and money and sex. That's all. Grounds for divorce. Okay. So that is the ciliated pseudostratified columnar epithelium found lining upper portions of the respiratory tract. Everybody good with that one? Okay. So I hope that what you are hearing is I want you to learn structure-function relationships with histology, because if you do, you're more likely to retain them than just looking at pictures. And my objective is always to try to get you as much retention as possible so that you are not completely confused when you get to physiology, or when you get to pathophysiology, or when you get into nursing school or med school. I would like for you to have as much retention as possible, because you're going to be more successful than other people if you have more retention of the things you've already been exposed to. Okay, so let's talk briefly about the surface features that we've been talking about. Okay, so cilia versus microvilli, we did that, right? Okay, so now hopefully you're getting to the point that you're a little more confident in recognizing cilia versus microvilli. Not perfect, but getting more confident. So I'm going to tell you that you can make your life a little easy because if you see a tissue and you're sure it has cilia, it has to be one of two tissues, doesn't it? What are they? Ciliated simple columnar epithelium, yes. And ciliated pseudostratified columnar epithelium. Yeah? Does that make sense? So if it's cilia, it's not 14 possibilities of tissue, it's 2, right? So you can like logic your way to it. It's got to be one of those 2, yes?
Sorry, I'm going to be the slow one. Will you repeat those 2 just one more time?
Ciliated pseudostratified columnar epithelium and ciliated simple columnar epithelium. Okay? And if you see really distinct goblet cells, and by the way, you can't depend on seeing them every damn time because they just aren't present in every single slice of every tissue. But if you see really distinct goblet cells, let me find you a tissue in which you can see them, then it has to be one of two tissues. Which two tissues dependably have distinct goblet cells? Ciliated, pseudostratified columnar epithelium, and? Not the other ciliated one, nope. And straight up simple columnar epithelium. That's the other place that you're going to see goblet cells. Simple columnar epithelium with microvilli. That's where you're going to see goblet cells. So here's a simple columnar epithelium with microvilli, and you've got like one or two goblet cells that you can see in there. Okay, so if you see a lot of goblet cells, you can assume it's one of those two, right? So you want to be able to like narrow it down, categorize it, have some logic, right, to work through it. Okay, so, and then last but not least, if you see distinct microvilli, it is always which tissue?
Intestines.
It is in the intestines. What's the name of the tissue?
Oh.
Simple.
Yeah, simple columnar epithelium. The straight up simple columnar epithelium is the one with microvilli and goblet cells. Got it? Okay. So what I'm trying to ask you to do is to stop just looking at pictures and start using a little like dichotomous key in your head when you're like, oh, I see this. It's got to be one of these things, right? And of course, that takes a little more exposure. But by the time you're getting close to a test, you should be able to do that. You're like, I know it's not this because of this. I know it's not this because of this, right? Good? Okay. So before I give you some pointers for studying, let's just do one last thing about epithelial tissues. Actually, yeah, one last thing about epithelial tissues, and that is simply to talk about how they are stuck to one another. And then we'll practice with some histology slides that you may or may not have seen before. Okay, so when you're talking about epithelial tissues, where the hell is that picture? Come here, picture. Okay. When you're talking about epithelial tissues, we need to, let me give you guys a metaphor here, okay? What I want to occur, and Lauren, I don't know what the heck you are, you won't fit into my metaphor streams here. These guys right here, on this front row, are all epithelial tissue, and you guys back there are all the cells in the connective tissue that is deep to the epithelial tissue. Question for you. What am I going to find right here? Basement membrane, yes? That is a group project between you and between them, right? That would be a basement membrane. Okay, so the metaphor that I want to use is epithelial tissue and its connective tissue is kind of like tiling the floor, right? You guys are the subfloor. We put down the adhesive, right? That was the basement membrane. And then we're going to put down the tiles, which are the cells. And of course, in epithelial tissues, the cells are right next to one another. But now I'm going to tell you that you're not done making a tile floor until you put in some grout, yes? Between the cells, yes? Okay. And the grout is what we call lateral cell junctions or lateral cell adhesions. And there's a couple of different ways you can stick these cells together. It depends on what you need. Just like tiling at home, sometimes the grout needs to be waterproof. Sometimes it needs to be like heat resistant, whatever, it depends on what you're trying to do. But here, there's three different possibilities. We can have a grout that is impermeable, meaning that if you guys are the epithelial tissue and I am something that wants to get into the deeper tissue, if you are impermeable, then I have to go through a cell and not between a cell, which gives you a lot more control over transport, right? Okay. So in order for that to occur, you would use a type of cell junction that is called a tight junction. And the tight junctions are these guys right here. What you would have to do with the tight junction is imagine a waterproof zipper, scuba zipper, that zips the two of them together. And it has to be three-dimensional because you can't make impermeability if you only do it at one point, right? Because then I could go under it, right? Okay. So they need to stick together in a way that I cannot get between them. I actually have to go through them, right? That's called a tight junction. Tight junctions are about impermeability. They're not necessarily strong because if I pulled on the side, I could probably pop them apart, but I can't go through. I'm trying to play Red Rover and they are bouncing me back. Okay? That makes sense? That's a tight junction. If these two weren't particularly concerned about impermeability, but they really, really wanted to be strong, then they would use a different type of cell junction. And that's the second one that you're seeing right here. This one. This one is called a desmosome. Imagine a spot weld between the two of them, okay? It wouldn't be impermeable, because if I was a toddler, I could go straight under it, right? If I were a gymnast, I could go straight over it. But it's really strong association, because what's going to happen is he will reach in and hold on to your side of skeleton, and you will reach in and hold on to his side of skeleton. So you got this really strong association. Desmosomes are about strength and resistance to stretching and twisting. So if I twisted them, they'd still hold together. It doesn't mean I can't get stuff in and over them. Does that make sense? Okay. You use that in locations where you need a lot of resistance to stretching and twisting. Your dermis of your skin has desmosomes. Okay. Even though the heart isn't, of course, epithelial tissue, the cells have to hold together despite the fact that you're constantly stretching on them and squishing them and stretching on them and squishing them. So that actually has desmosomes. Ladies, the cervix, like nostril size, right? You have to get a freaking cantaloupe through it and then ever use it again, right? I'd put some desmosomes in there, wouldn't you? Right? Hold that damn thing back together, okay? All right, and then the last ones are this strange type of grout that allows for communications between the two cells. So if they had this type of cell junction, what would happen is he would have a channel on his cell membrane, and he would attach it to a channel on her cell membrane, with me, so they could have almost instantaneous communication between them. Does that make sense? Okay. Those are called gap junctions, and that's the third one. They are not strong and they are not impermeable, but they do allow for almost instantaneous communication between cells.
A wired connection.
Yes, exactly, a wired connection. So what would happen is if you guys were all stuck together with gap junctions, right, you could make the whole group of cells sort of behave as a unit. Is that okay? So imagine that that's the upper portion of the respiratory tract. Here's the bottom portion of the respiratory tract. She lives in Salt Lake City like the rest of us, and so she's breathing in funk, right? What I want to do is I want to stimulate this bottom cell to start a wave-like movement of cilia toward the top, right? So what's going to happen? Engage with me right here. You guys are all going to do the wave for me. Do the wave. But then yeah, there we go, right? Okay, they're like, damn it, I don't want to do this. But what's going to happen, right? Peer pressure. Okay, so that is what gap junctions are for. Now, do you have to use just one? You don't have to use just one. You could use multiple different kinds. If you needed both strength and impermeability like this, because we don't want to lose water, we would use two. What? Tight junctions and desmosomes. If, like the heart, you needed both strength and communication to allow them to work together, you would use desmosomes and gap junctions, right? So there's no rule that says you couldn't use more than one. Those are the lateral cell junctions. Okay, so how do you know when you have studied epithelial tissue enough? Don't do what most students do, which is just name tissues, move on, name tissues, move on, because that's not all the questions that anybody could ask you, either in lecture or lab. So here are the questions that are reasonable questions that you could be asked about any epithelial tissues, and then we'll practice them a little bit, okay? So when you see any epithelial tissue, the first thing is you should be able to identify that it's an epithelium, right? Which of the major categories of tissues does this fit into? And you guys are really good at that right now. You're like, it's got an apical surface that's free. I know it's an epithelium, right? And then most students do the next one, which is I can identify the name of this tissue. What is it? Stratified cuboidal epithelium. But what they don't do is go further. And there are other reasonable questions to ask, and that is there any modified structure? For instance, what's that? That's a lumen. What is C? That's cilia. What's B? Goblet cell, right? Any modified structure is fair game. Anything that you ever learned the name of is fair game, right? Okay, so what about surfaces? So what's the surface? Apical surface. What's this surface? Basal surface. What is this structure? Hold on. What is this structure? Basement membrane. If I ask for a surface, it's basal. Yeah? Structure, right? Different noun. Asking for something different. It's basement membrane. What was your question?
No, you answered it.
Oh, okay. There we go. Yeah, that's really part of the onus in anatomy and in phys is I have to write a clear question or else I'm going to get all kinds of garbage as an answer, right? Luckily, this ain't my first rodeo, and most of my bad questions have been revised by now. It doesn't mean that I won't do it in the future, but I try really hard to be very clear because I don't want to confuse you when I'm writing questions. Okay, and then apical surface, basement membrane, specific body locations. So let's practice a little bit. I know you guys love specific body locations. It's your fave. Okay, so a specific body location for this tissue with the red. Yeah, the alveoli of the lungs. Anything else? Nope. This is what tissue, by the way? Simple squamous epithelium. Lining blood vessels, right? How about a specific body location for this tissue? Yeah, the lining of the mouth. That's great. How about a specific body location for this tissue? Lining the male urethra. Lining the male urethra, right? So that's going to take a little practice. And again, you don't have to memorize every body location. You need to find a true one that you can remember so that you can write it down. But if it's only found in one location, that's fair, right? I can put male urethra on a multiple choice question because that's like the only place that it's found. The other ones will probably be either something that I said aloud in class, which is always fair game, right? Or a fill in the blank question, right? Okay, good. So this is what the good student of histology would do. Instead of just looking at a histology image and naming the tissue, look at multiple histology images, force yourself to answer all of these questions, and then go on to the next one. And then there really won't be any left field questions for you either in lecture or in lab. Oh, the last little thing is don't forget that these are like cells. So it's not outrageous that somebody could ask you what that was, right? That's a nucleus, right? Right? And there's like some intracellular fluid or cytoplasm in there, and there's like some cell membranes, and that's all reasonable for you to understand and could be a question. Good? Are we good? Okay. All right, so last thing, and you don't learn histology slides for these just because they're really messy and hard to identify. We have been doing covering epithelium, like things found on surfaces. But just so you know, all of the glands in your body are made of epithelial tissue as well, even if they're not on surfaces, right? And glands aren't always associated with surfaces. Like the pituitary gland ain't nowhere near the surface, right? Okay. So first off, glands are all made of epithelium. And like we mentioned, I think we mentioned this in this class last time, it is actually clinically significant that glands are epithelial tissue because, of course, epithelial tissue has a relatively fast rate of cell division and regeneration. And the stats are just not with you as far as cancer. If you are dividing all the time, then it's more likely that you are going to lose control of cell division. So both glandular epithelium and surface epithelium both have higher rates of cancer than all of the other 4 categories of tissues. So that ends up being important. So what is a gland? A gland is one or more cells that produces and secretes a product, right? So that means, of course, that the simplest gland you can think of is a goblet cell. That's a gland, right? It's a unicellular gland. And now we have to define glands as endocrine or exocrine. And that is not quite what it implies. It's similar to it. You would think that it would be in versus out, but it's kind of not exactly that. An exocrine gland will secrete its products onto a surface. And the surface doesn't have to be external, right? It just has to go onto a surface. For instance, exocrine glands include salivary glands and also digestive glands, right, that secrete onto the surface of my digestive tract. Does that make sense? So it's not necessarily in versus out, but it's always toward a surface. But endocrine glands do not secrete something onto a surface. They secrete something and it gets picked up by the bloodstream, right? And they specifically secrete hormones. That's what endocrine glands secrete. They look like a mess when you try to look at them in a histology slide, so I'm not going to make you memorize what endocrine glands look like. But let's talk about endocrine versus exocrine for just a minute. So exocrine glands can either be unicellular, like a goblet cell. So, by the way, with that categorization of secreting onto a surface, isn't a goblet cell an exocrine gland? Yeah, it's going onto a surface, right? Its product's going onto a surface. But then there are also multicellular exocrine glands. And no, you do not have to memorize all of these. You just need to be able to recognize that they are epithelial tissue and exocrine. And how can you do that? Well, exocrine basically, doesn't this look like an epithelial tissue? Yeah, doesn't it? Right? It does, right? And doesn't it have a surface? It's got a duct, right, that leads to a surface, right? And it is epithelial tissue, so it is exocrine. Yeah? Does that make sense? Now, a duct from a top view usually looks like a pore or a hole. Yeah? So let's talk about a few glands and see if you can categorize these, okay? Endocrine or exocrine. And by the way, in my class, you don't have to name these individually. You just have to be able to recognize that they are epithelial and exocrine. That's it, okay? So categorize these glands as endocrine or exocrine. You ready? Lacrimal glands secrete tears. Exocrine. Thyroid gland. Endocrine. Yeah. Pituitary gland. Endocrine. Mammary glands. Exocrine, right? Digestive glands. Exocrine. Yeah. Exocrine that dump into the digestive tract. Does that make sense to you? Okay. So endocrine or exocrine just has to do with where their product ends up. Is that okay? So that's endocrine versus exocrine. And of course, in physiology, you do tons and tons of endocrinology. You don't really do very much of it in anatomy at all, just because it's not really an anatomical system. It's more of a physiological system. So you've got exocrine and endocrine glands. You will notice that by talking about those, you also probably thought, oh, a lot of those actually get cancer, and they do, right? How about sweat glands and oil glands? Exocrine. Sweat glands and oil glands are exocrine as well. Okay, everybody good? Endocrine versus exocrine. For these, you do not have to know histology slides. Just have to know that they fit into the categories that they fit into. Good? Okay, so now let's talk about connective tissues. You ready? Okay, connective tissues, and I'll just go ahead and redraw this for you guys. Connective tissues, for me and for many students, are the most difficult of the four categories. And I think it's for a couple of reasons. First off, they're really, really variable, both in structure and in function. So that makes them a little harder, because like there's no really easily recognizable pattern like there was with epithelial tissues. Really variable in structure and function, and nobody decided on a naming convention. So what they're called is whatever the hell somebody decided that they were called, right? So there's no particular naming convention. There is a way to organize your knowledge a little bit so that you get a little better at it. So where are they? Where are they? They're anywhere that the other three aren't. If you don't need to wiggle, you're not talking about the brain or the spinal cord or nerves, and you're not talking about surfaces, then it's connective tissue, right? And it's as broad as bone is a connective tissue, and fat is a connective tissue, and blood is a connective tissue. So the question that comes up is, why the hell do all of those weirdly different tissues fit into the same category? And although this doesn't particularly help you, at least it might tell you why. The reason that they all fit into the same category of tissues is because they're all derived from the same embryonic tissue. So they're all derived from the same embryonic tissue. And that embryonic tissue, we'll look at it next time. It doesn't look very impressive. But that embryonic tissue is called mesenchyme. And from mesenchyme, all of those really different tissues arise, right? So blood and bone and cartilage and fat and all of that stuff arise from mesenchyme, so they're all categorized that way. Okay, so what is true, and we're going to do the categorization in just a second. What is true generally, and there's always exceptions, what is generally true about a connective tissue is that the cells are usually more widely spaced than epithelial tissue, yeah? And of course it doesn't have any surfaces because it's not at a surface, right? So it's not going to have like a free surface. So whereas epithelial tissue, they were right next to one another and had a free surface, these cells are going to be widely scattered and have no free surface associated with it, right? So widely scattered cells tends to be true of connective tissue. And then there's a bunch of stuff between the cells that you're going to have to label. And the stuff is called matrix. And then it's got some fibers in it, some other stuff's in it, and we'll get to it next time. So lots of widely scattered cells. The other thing that's true about connective tissue is that it is usually more vascular than epithelial tissue. Not every single part of every single connective tissue, but generally there are going to be blood vessels going through it. So widely scattered, lots of space, usually vascular. So how do you sort of subdivide so you can keep all of these tissues organized? Well, the subdivision of these, I always lose my pen. Okay, thanks. Constantly doing this. Okay, ready? So they're all derived from mesenchyme. That's the embryonic tissue. And then as an adult, you are going to have 3 big categories of adult connective tissue. All of these are adult connective tissues. Let's do the easy ones first. There is one that's called a fluid connective tissue, and it's the only one that you learn. Okay, the only fluid tissue that you learn is what?
Blood.
Blood. Yes. So you only learn one, and it's blood, and it's pretty damn easy to identify, honestly. Like, yep, that's blood, still blood. Good. Okay. And then the next one is the opposite end of the spectrum of blood. These are your most solid tissues. They're called supporting connective tissues. They resist gravity and help make you stand up so you're not a flesh-colored pancake. And these are supporting connective tissues. And they include what and what? Bone. And you only learn one bone slide now. There are actually 2 that you will learn eventually. But for right now, you only learn one bone slide, right? And then how many cartilages do you learn? Do you know?
3.
Yeah, there's 3. We'll learn 3 cartilages. Good? And then last but not least, and this is the one that gives people the most trouble because there's the most of them, and it's a little hard to organize, is these are called connective tissues proper. And if this is fluid and this is mostly solid, this is the squishy stuff that is in between, and it's most abundant, right? And when you get to the squishy stuff, there's a further subdivision, connective tissue proper. There are loose and dense. Okay, and the loose versus the dense has to do with looking at the matrix and saying, there's a whole bunch of fibers in there or not very many. So even if you can't quite do it yet, I just want you to see if you can tell a loose from a dense connective tissue. So you ready? What do you think this one is? Loose. What do you think this one is? Dense. Okay, so let's look at a few more and see if you can do those loose versus dense. And then you'll start getting used to it. Okay, hold on. Connective tissues. Okay, so when you're in the propers, you have to make sure that you're actually in the propers. Let's talk about this one. Yeah, that's dense, right? Do you see? So it's just got to do with how many fibers you actually see in the matrix. Let's do another one. How about this one? Yeah, that's loose, right? So loose versus dense. Does that make sense to you guys? Okay, that's what the loose versus dense has to do with it. And by the time we get finished, and we'll do this on Tuesday when I see you, there are 3 loose and 3 dense. Good? So by the time you get finished, you will learn 3, 6, 9, 10, 11 connective tissues. Good? Now, why do I focus on stuff like this when in lab they don't make you categorize them? Because this helps with recall. Because if you have a place to put something and you're like, I can't remember what that one's called, but it looks like a loose. There were 3 loose. It's not this one or this one. Then you pull up the name, right? It also helps with long-term retention. FYI, just a little bit about me. I'm an anti-memorizer. I'm an always fit things into some logic or category because memorization, just me with my own particular brain, it doesn't stick with me very long unless I have context for it. So you are taking a class from someone who is trying to make you avoid rote memorization. If that's not your thing, sorry. It tends to not stick with you very long, and I teach you guys in phys, and I want you to remember. It helps me when you guys remember. It also will really help you. So I do a lot of categorization and a lot of organization, and sometimes they don't do that stuff in lab, but it's all right. It complements what you do. Okay, so what are some functions of connective tissue, and then I'll let you guys go. Connective tissue does damn near everything that the other tissues don't. So you're not going to find it on the surface. Like, you're not going to find connective tissue on the surface unless you've had a really crappy day and you got a bone sticking out, right? Okay? And you're not going to find it wiggling anything, right? Connective tissue is not the source of wiggles. That's muscles, right? It's not going to be sending electrical signals, of course, because that's nervous tissue. But so broad functions anything else that you could imagine. It is sometimes protective. Sometimes bone's primary job is that it is protecting you. Name me a couple places where bone is even more protective than it is about, for instance, leverage for movement. Yeah, ribs and cranium, right? Yeah, you got some muscles stuck there, but not very many, right? It's not that wiggly compared to other parts of your body. Some places where fat is protective. What? It's actually not that protective against in your abdomen, because you know super duper hot people, they're just fine, right? Right? Somebody who's got an 8-pack up there, their abdomen is not less protected than yours. They're all right. Where do you need fat? Protective. What? Kidneys, heart, those kind of locations, sometimes in your joints for padding, sometimes fat's protective. Let's just all be honest. We're Americans, and we should know that we do not need this kind of protection on our thighs or our butt or our gut. It's just there for a rainy day. So what about slapping things to other things? Tendons and ligaments do that. Do you know the difference between a tendon and a ligament?
That's what it attaches to.
Yeah.
They both go to bone, but which one's which? Which one's which? That's all right if you don't know. We're in the process of learning. Tendon is muscle to bone, and a ligament is bone to bone. But they're the same tissue in two different sort of places in the body. Okay, storage of fat. Not only does your adipose store fat, but your bone marrow also stores fat. And basically, fat is quite often about nutrient storage, energy storage for later, right? Disease fighting. Blood is a connective tissue, so every damn thing that blood does is a connective tissue function. And you could spend an entire course talking about what blood does, right? Okay. And then last but not least, sometimes connective tissues produce other connective tissues. For instance, bone marrow, a connective tissue, produces blood, another connective tissue, right? So is this a comprehensive list of connective tissue functions? No, of course not, right? It's just like, hey, it does a lot of things. Look at all the stuff that it does, right? That's what we're trying to accomplish here. Okay, so on Tuesday you do not have a quiz, but next Thursday you will have a histology quiz. So you're going to be working on histology. You can go ahead and get a little ahead because we'll do connective tissues and hopefully the rest on Tuesday. Cool? Okay, have a good long weekend. I think, by the way, that no Monday open labs because the college is closed, but Friday and Saturday open labs are still a go. Yeah.
Right, you said something about figuring a way for me
Hold on,
to
let me.